Hello, everybody. Welcome to the IWGSC webinar. We have a great webinar today. My name is Joshua Stein. I'm the project manager at IWGSC. In just a few moments, we'll be joined by Simon Griffith, and so big treat today. Before we start, I wanted to give a little bit of an introduction about the IWGSC. As you may know, we're an international consortium formed about 20 years ago with the goal of sequencing hexaploid wheat, which was, as you probably know, was accomplished by 2018. Uh, Today, the consortium consists of about 3,500 general members um, coming from 971 institutes or companies in 80 countries. And I particularly want to thank our sponsors who make events like this possible. So I am going to thank them here. We have Arbor Biosciences, INRAE, CIMMIT, RAGT, Kansas Wheat, and Florimond Desprez. So thank you so much to our sponsors for making this webinar possible. As I mentioned, the IWGSC, our biggest role in the community was to bring a reference genome sequence to the community. Today, our 2.0 vision is to enhance breeding through an increased understanding of the molecular basis of traits and their allelic diversity. And in order to accomplish this vision, we now have a funded project, the IWGSC Wheat Diversity Project, with the goal to preserve, discover, and mobilize genetic resources by sequencing genomes that encompass the worldwide diversity of bred wheat. And in this effort, we are providing platinum assemblies for 12 maximally diverse wheat landrace accessions. And we are also updating Chinese spring to a platinum sequence level. So that would be RefSeq version 3 is what we're currently working on. In addition to that, we are doing some transcriptome sequencing across these accessions to provide evidence for gene annotation. We plan to update the Chinese Spring RefSeq as well, and we want to retain continuity between previous versions and the new version so that people can make that transition. And then finally, these data are going to be represented as a practical haplotype graph by our colleagues and collaborators. And I wanted to acknowledge funding from the National Science Foundation and the European Union on this project. So stay tuned and we'll have more information as this project goes forward. I want to remind people we have our next upcoming webinar will be on July 17th, and we'll be joined by Agnes Sabo-Haver of North Dakota State. So there's a registration here. So please, if you can make it, please register for this for our next webinar. Okay. I just want to take a very quick moment to introduce you to the interface, the webinar interface. If you have questions during the talk, you can type your question into the question and answer box. That will be saved for the end of the once Simon has completed his talk. We will then go to those questions. There is also a chat window, and that is used to communicate information, for example, providing handouts or other things, or if there are technical issues that we need to communicate to our audience, we'll use that chat. Finally, I want to remind people that this webinar is being recorded, and we will be posting it on the IWGSC YouTube channel, so you can always rewatch it. The reruns are really great to watch. Okay. Okay, finally, I want to introduce our presenter, Simon Griffith. He is the group leader at the John Innes Center in the UK, and the title of his talk is Breeding: Why We Need to Start All Over Again. And with that, thank you for your participation, and I will bring Simon up. Thank you so much.
Thanks a lot, Josh. I'll just start sharing. Okay, do we have the presentation?
I don't see it yet.
Oh, what's happened? No?
Yes.
Great.
And if you can make it bigger in any way, or if that's as big as it is, that's fine.
Currently it's full screen. Maybe I've gone
Okay.
on.
Okay.
I will stop sharing. Sorry. Better?
Okay. Looks good.
Oh, good. Thanks very much for that kind introduction. It's a pleasure to be speaking today for the IWGSC. So speaking about breeding and starting points in breeding and maybe going back a little bit, I thought I would begin by talking about, just going to pointer, where breeding really began. And that was, of course, in the Fertile Crescent. So we began 10,000 years ago in these first cities. This is the oldest city in the world, Çatalhöyük in Turkey. And you can see this is a grain store in one of the houses. And whenever you go back to these first cities, you see granaries, grain stores, and it really brings it home that wheat and in other centers of domestication, other crops like maize and rice, were absolutely pivotal and central to the establishment of civilization. And of course, this changed human society totally. With the establishment of these cities, the world population increased rapidly. And at this time, in the Fertile Crescent, the world population went up from 1 million to 10 million. And then really, I think that's helpful to think about like that because nothing's changed. The world's cities still rely on these few crops, and wheat is still an absolute staple pillar of the global society. So that's currently for a population of 8 billion people. We often hear it said we're going to have 10 billion by 2050. And even if we carry on managing most of this orange line is human population, and the blue line is wheat production. And wheat production is a great way of predicting human population size, right? It's highly correlated. And let's assume that through genetic gains in breeding, because that's what's delivered most of these increases, we can carry on following the population increase right up to 2050. Even if we can do that, we still got the problem that wheat and agriculture generally is a major contributor to climate change. And increasingly, most of the greenhouse gases will come from agriculture. That's the orange line, sorry, yellow. Meanwhile, agriculture is removing ecosystems, and the use of agrochemicals is having massive impacts on biodiversity. So the grey line is the Living Planet Biodiversity Index. So we face these huge challenges in agriculture, and that's why I don't think it's too bombastic a title to say things like we need to restart breeding again, because the challenges are enormous. So again, thinking about where do things come from? After 9,900 years of wheat being spread around the world as landraces has passed between farmer and farmer and improved in a probably an unconscious way, systematic wheat breeding took hold in the early 20th century. And this is probably Britain's kind of father of breeding, which is Roland Biffin. I'm sitting here in the Biffin building at the moment. And Biffin's receiving a prize from farmers in the county of Essex who are generally incredulous, and you can see from this newspaper article that they talked about their problems of yellow rust, poor bread making quality, and Biffin has taken the relatively newly discovered Mendelian rules and used them to achieve these improvements. Fantastic stuff. And that's in 1921. He's receiving his prize. But one consequence of this systematic plant breeding, the type of breeding that Biffin started in Britain, is that pedigrees go from being multicolored, where landraces and non-adapted varieties come from many different sources, to monotone, where actually in breeding for a mature breeding system for one target environment, you end up increasingly crossing well-adapted with well-adapted and the material from your own breeding pool. So necessarily there's a reduction in genetic diversity. And increasingly there's evidence that we can begin to hit glass ceilings with the diversity that's available. Again, these are UK yield increases in the UK, spectacular through the 60s and 70s as a result of the Green Revolution, good in the 80s, but then just in the case of grain yield, in this case, plateauing from the 90s onwards. So considering a lot of those gains came from breeding and the shuffling of genetic diversity, we could say, well, maybe we need to tap into that diversity that was lost again and go back to use it. Many ways you could do that. Great groups all around the world using different sources of diversity. But the thing that I started doing about 15 years ago was to take a landrace collection called the Watkins collection. This was exactly at the same time that Biffin was receiving that prize. Watkins was sending out letters around the British Empire, requesting seed from different, through the London Board of Trade, for example, to embassies, consulates, army barracks, anything he could do to get some seed recovered from local markets and sent back. If you're interested in the Watkins story, it's a really nice story. This article in the Genetic Society magazine by Robert Koebner digs into Watkins' life and how he came to be doing this, and I really recommend it. He was quite reclusive, and actually that's the only photograph we can find of him leaning out of the window at the University of Cambridge. He wouldn't even join the departmental photograph as one of the main group. These are the kind of letters he sent out, and we've digitized these now, and we're trying to get them online. They're really wonderful documents, a whole set of documents to, as exactly as I describe, to build his collection, letters to Vavilov in the USSR, etc. And in this one, it's a typical template which he'd send out to the collectors, in this case going to Italy, Sicily, Corsica, Romania, and Algeria. And in the letter, Watkins is saying, we want to make a global wheat collection. And we've seen from the work of systematic breeding at the Plant Breeding Institute that that's going to take over all around the world. And these valuable cultivars are going to be displaced. So he says in this middle part, we don't want the improved stuff. We want you to go to distant parts of the country, to markets, and collect the wheat as it is now, because it's going to be displaced. And finally, he says exactly the same thing here. Don't go to these newly established plant breeding stations, go out around the country. So a real visionary, in my opinion. The collection as it stands today, it's not the same, but as big as the collection that Watkins originally assembled due to the ravages of time. But now the wheat collection, the bread wheat collection, is 827. And these are the countries of origin. Now, our basic question, to put it really simply, is when Watkins did this, did he preserve alleles that are missing in modern wheat? It's really simple. And the simple answer, on paper at least, is just say, well, sequence it all. Sequence modern wheat, sequence the Watkins collection, assemble it to the fantastic reference genomes and say, yeah, what's the missing part? Are we totally overlapping and breeders did a wonderful job of sampling everything? Or is there a lot of diversity which is absent? Of course, that type of work is both technologically and financially challenging. So in terms of the technological expertise, the bioinformatics clout, and the collaboration, I was delighted to build up a collaboration with Shifeng Cheng at the Agricultural Genomics Institute of Shenzhen. And we just took that simple approach. We're going to sequence the entire collection, shotgun sequencing to 13x. And then we're also going to sequence a modern sample of wheat. We took this from Axiom genotyping, and at the University of Bristol, took the maximum diversity of modern wheat assessed by Axiom high density genotyping. And we'll just compare the two. And these are the results of that. This is a complex slide, but it's the only genomics I'm really going to trouble you with. And if you look at first of all, when we sequenced the Watkins collection, we structured it into ancestral groups. So ancient populations that reflect the first spread of wheat from the Fertile Crescent. The ancestral groups are color-coded in this map, and you squint, you'll see that they're associated with geography, I suppose, unsurprisingly. And then this brick red color, this is our modern wheat sample and where it comes from. And then when we do a kind of a relationship analysis, a T-SNE analysis in this case, you see that modern wheat is overlapping with two of those ancestral groups, ancestral group 2 and 5. These are two of the European groups. While in fact, the remaining 5 ancestral groups of the Watkins collection are phylogenetically isolated. Modern breeding did not use this diversity, and it's been left behind in the countries it came from. This is because the advent of systematic breeding was highly Eurocentric. It happened in Germany and France, and then later in Britain. And these ancestral groups, the green one is kind of Central Eastern Europe, and the purple is basically France, Western Europe. And you can see here the red colors are the pangenome references sequenced, and they're in the brick red color. So we really haven't touched on this diversity. It's in the Watkins collection and other landrace collections around the world. And it's not just a matter of shuffling genes to see different haplotypes. The blue colors here are SNPs, indels, CNVs, copy number variation and haplotypes, where blue means it's only in the Watkins collection. In every case, that's over 60%. So the vast majority of SNP diversity, for example, is not present in modern wheat. And then together with Xiaoming Wang and Christobel War, we took on long-range haplotype analysis. This is the whole of the wheat genome, with modern varieties going from the 20s to the early 20th century. And this purple and green color, this is ancestral group 2 and 5 again, right? It's not scrambled quite the way you might have expected as a mosaic. Multi-hundred megabase segments of modern wheat are just those same landrace donors. And you can actually rebuild modern wheat over 50% of the genome from just 26 of the Watkins landraces, just from ancestral group 2 and 5. So the bottleneck, I reckon, was tighter than we ever thought. That's just genotypic diversity. We need to associate with phenotypic diversity to do anything useful in breeding. The obvious thing in GWAS, and this whole population is incredibly good fun and great to work on with GWAS. This is one example here in collaboration with JC and Rothamsted, led by Rothamsted really, where we're identifying genes for zinc content, and that's an appropriate thing to do. But in many, many cases, GWAS is a weak tool for dissecting diversity such as this, and there's two reasons really. One is that those many alleles that are not present in modern wheat that I talked about, many of them are present at low frequency, and GWAS is not good at detecting low frequency allelic effects. Secondly, the collection itself, you'll see the string and metal rods here at Rothamsted when they're growing the Watkins collection. Hundreds of kilometers of string. Landraces are difficult to work with. They don't stand up. And physiologically, the context of the expression of the traits is not the same as it is in modern wheat. So it's important that we assess those traits in a modern context. And we did that by piggybacking on the maize, innovations in maize genetics, and making nested association mapping populations. So there's a nested association mapping all based on Paragon, which from Watkins and other genomes we use now includes 12,000 recombinant inbred lines. And then to map the traits using GWAS, QTL analysis, and nested association mapping, we built an integrated program in the UK. It's the UK wheat program, currently DSW, Delivering Sustainable Wheat, but it's been Designing Future Wheat and the Wheat Institute Strategic Program, and it's cross-institutional. So between our institutes, and these are plots at Rothamsted, we began to dissect the phenotypes and map them back to the genetic variation. So altogether, we measured 137 traits, and they're for things like biomass partitioning, phenology and adaptation, yield components, both in terms of biomass and numeric components, the nutritional qualities of the grain, protein, mineral quality, mineral quantity, etc. And also nitrogen partitioning and nitrogen use efficiency. Taking that trait set together, we did a really extensive QTL analysis and GWAS analysis. And I'm not going to overload you with that stuff today, but I'm going to give you a high-level overview. So using those three methods, for example, for GWAS, and here's just one example. This is winter kill in Shandong, the major effects for winter kill. But this isn't just winter kill MTA, this is all the market traits associations. We see 1,428 MTA. We identify QTL, in this case 3,280, and using NAM-GWAS, which in a way is the most spectacular thing for me. This plot, underneath this plot, is 6,000 recombinant inbred lines, and it allows you to completely dissect flowering time of wheat, landing on all the major genes, PPD, VRN, FD1, etc., but also a few others and allelic series within them. So with using NAM-GWAS, we have 3,545 genetic effects identified. Again, so we're getting there. This is associating traits with genotypes, but are they useful in modern breeding? We sieved them down to the ones where the increasing effect was in the desirable direction. And the haplotypes, so these molecular signatures on the DNA, showed that the haplotypes were not present in modern wheat. And that still gave us hundreds of new effects, which were beneficial for modern wheat, but not present. And you get striking results. This is that yellow rust QTL example expanded. Just one yellow rust, major yellow rust gene on 2B. And we looked at where these yellow rust resistances come from. These are all to the warrior race, so the most pathogenic recent race in the UK. And you can see by the color coding here, they're all centered on Iran. And you couldn't have a better example of, remember I told you those ancestral groups were Western European and Eastern European, nothing, right? No resistance to the warrior race. And none of those breeders were dipping into these gene pools, but that's where the yellow rust resistance that they need now is located. To give you some vignettes of the type of things people are doing with this is Petros Sigalis leading work on mineral analysis in the Watkins collection. And he produced something called the Mineral Atlas, and many major effect QTL for beneficial nutrient alleles again. This is work on dietary fiber, so increasing fiber in the endosperm. And although we'd already identified the QTL for these in elite wheat, what you find in Watkins is an allelic series. So these increase arabinoxylin and dietary fiber to an even greater extent. Thinking about adaptation to climate change and temperature sensitivity, there's a Swiss National Science Fellow in my group called Lucas Cronenberg. And Lucas used the field phenomics facilities and the isogenic lines that we produced from those novel QTL, when present in modern wheat, but had beneficial effects. And he's dissected the ambient temperature response, so you can start to rebuild ambient temperature response in varieties. And traits which were completely intractable, at least in the UK in modern wheat. This is the grey field slug, screened the Watkins collection. We found significant resistance to grey slug damage. And now we've mapped that as a QTL and we're trying to introduce it into breeding material. The more normal trait, the one that is actually the most of the entries for our breeding toolkit, which is the way we move these things into commercial breeding, are actually yield. And this is work from Christopher Wawi and James Simmons up here, where they've identified new yield effects, in this case through 1,000 grain weight, and the unique Watkins haplotypes give a yield increase. This proper full-scale yield plots on multiple sites over and above the haplotypes which are in modern wheat now. We move these things into modern wheat. It's through a back crossing process. And we've gone from all those only ancestral group 2 and 5 to a multicolored kind of introgressome in one genetic background. And this gets by the way, I want to say all our material is deposited in our gene bank, the germplasm resource unit. And everything I talk about today is available on request with a and MTA, etc., which should give you full freedom to operate. All the germplasm ends up in the germplasm resource unit led by Noam Hayoud. And then for this breeder's toolkit, these are the isogenic lines that we made from the prioritized QTL, unique to modern, absent in modern breeding. We then collaborate with all these commercial companies. In the UK, a lot of this research is public, but all the breeding is commercial. So these 7 companies accept this material and actually vote it into their plots. And then we grow fully replicated, usually yield plots, because that's usually the trait we're assessing on all of these sites. And this is where the breeders take or leave those outputs that we've produced. We're really proud of these achievements and we think they're effective. When we do anonymous surveys, the breeders tell us that they're moving into their pedigrees. And actually there are varieties appearing in the European national lists now, which have pedigree from the UK wheat program. But if you think about everything I've just said to you, that actually it's not tens to hundreds of QTL that are important that we introduce into modern breeding, it's actually thousands. And many of them are small effect. They're not these large effect QTLs, but they're small effects, which when added together with modern breeding techniques and selection, would actually contribute a hell of a lot to the genetic gains we make in breeding. You can't back cross in thousands of genes. You've got no genome left of the original recipient parent. So you need an alternative approach, and that's what I'll talk about now. This summarizes what I've just said again. These are the ancestral groups. This is 2 and 5. What modern breeders did was took what was near to them, the lines that were selected in the 19th century probably, and bred with them, I think in France and Germany. Then they didn't really respect those ancestral groups. They just intercrossed with them, and you saw them in the long-range haplotypes from Cristobal. It's mixed green and purple. And then they imposed empirical selection on them in classic pedigree breeding. So they're breeding for adaptation, reduced height, yield, of course, bread making quality, and disease resistance on the whole. But I would make the point on this that we often think of breeding as kind of progressive and incremental, and every year it's getting a bit better. But a lot of this breeding is kind of circular. It's maintenance breeding. You're having to breed for resistance genes that have broken down, or you're having to breed for an environment which is actually changing due to climate change. So you don't need 100 years to get to a variety that you're at today. And then that's even more the case when you think about the technological advances that the breeders, the forefather breeders, didn't have. So you see here, I've just highlighted on the right, line selection, Mendelian genetics, the Haber-Bosch process, statistical genetics, mechanization, the Green Revolution semi-dwarf genes, then marker-assisted selection, and now only in very recent years, genomic selection, genomics itself, the work of the IWGSC, and gene editing, absolute revolutions, which haven't been applied to wheat breeding throughout this process. So maybe we can start again, and with this knowledge, really accelerate what can be achieved from the beginning, which is what this slide signifies. But it's not the first time us or anybody else has tried this. And if you cross 2 Watkins accessions together, or only 2 landraces, what you get is a horrible mess. This is a picture of landraces growing in the field, Watkins growing in the field without stakes and string. And when you cross them, it gets even worse. They actually get taller, I guess, through heterosis and the association of various alleles. And it's impossible to move a pedigree forward that way. So what do? Well, with gene editing in mind, we could say, well, let's fix all the genes we think we need, the things that you could describe as must-haves in the pedigree. That now that after years of careful and difficult breeding, these alleles are actually fixed. And you'll see it in different environments. If you go to the USA or the Mediterranean, you'll see different combinations of these genes which are fixed in the right combination. And chief amongst these, and the one I'm going to really focus on today, is the major height gene RHT1. But we're also working in the same way for your information, and please do follow up with me if you're interested, on the less talked about Green Revolution, PPD1, Green Revolution gene. This is the photoperiod sensitivity, where you're tweaking the response to the wheat is usually a long day plant, but this can make it, depending on the allele, day length insensitive or more day length requiring. In the wheat, we want it to be late. In the UK, you want it to be late photoperiod sensitive. Grain color. All these are clone genes. So this is the R gene, a MIB factor. And in the UK, you need red grain color to reduce sprouting. Whereas if you're in an environment where sprouting is not a problem, white grain is actually quite desirable. And if you're going to make bread out of your wheat, you need it to be hard milling texture. And these are the purindolines, and deletion of the purindolines makes hard milling wheat. So we're saying, let's fix all these the way we want. We want hard, red, late, short wheat. And then we don't have to select for these things so much. We can focus on all those thousands of allelic traits and allelic variation in the Watkins collection, which is the new stuff. So this kind of makes a framework for them to hang on. A little bit like the sequencing, of course, that's always something you'd know you'd like to do, but wasn't possible. It became possible conceptually with the invention of Cas9 CRISPR. It became politically more favorable, at least in the UK, with the passing of the Genetic Technology Precision Breeding Bill, which has recently had has been fully passed in the House of Lords. Gives us more freedom to innovate in this area. And then also we've been lucky enough to receive some funding. So everything I've just described was my main funder, the BBSRC in the UK. What I'm going to describe from now on is actually funded by a government department called the Department of Environment, Food and Rural Affairs, and they fund this thing called the Wheat Genetic Improvement Network. So to carry out this dream of fixing the alleles we want in the land races, we needed to transform the land races. Again, only a few years ago, we were only transforming single genotypes of wheat, things like Fielder. With the innovation of Juan de Bernardi and others at Davis, and building on that from other research in developmental genes, this GIF-GRF construct, protein fusion, actually massively increases the repertoire of genotypes that we can transform. And that's a platform that gets ran in the wheat transformation platform here. But here, Rajni Awal has taken Watkins accessions and seen what she can transform. And in the tests with Gus here, she found that she could transform 6 Watkins lines with an efficiency between 4 and 33%. So we were good to go. We'll start our gene editing experiments. As I've said to you, the first one that we wanted to really try out was RHT1. I'll tell you a little bit about the way this gene works. Many will know, I'm sure, but it's the allele that's used in the Green Revolution confers insensitivity to gibberellic acid. So normally when you put gibberellic acid on a dwarf, it rescues it because the GA pathway has been knocked out. Put GA on any wheat plant, it gets taller. But in the case of this gene, it actually reduces that sensitivity. This is a GA signaling molecule, and it works it's actually a growth repressor. And when GA interacts with this DELA domain, you release the repression. So that's when wheat can grow. So in the Green Revolution, the repression is always on because this DELA domain is actually removed from the protein. The way it's removed is with a stop codon just after the DELA domain. And that means the protein translation reinitiates at a methionine just after that stop here. So there's 3 ways you could make these alleles with gene editing, 3 obvious ways. One would be to delete the DELA domain, which is, I'd say, we're going up in scale of difficulty here. That's relatively routine in gene editing laboratories. The second would be to do something which is more preferable, which is to recreate the natural allele, the natural variation. You can do that with base editing. So in RHTB1b, this green color here, this stop codon is the conversion of this C to a T. And it's feasible that base editors could make that change for us. But the main Green Revolution allele used in the UK for historical reasons, is OHT D1B. You can't use editors to make this change, but you could use prime editing. But prime editing has actually so far been highly inefficient in plants, and certainly in wheat, to our knowledge, not being achieved. So there are like our 3 levels of difficulty, 3 things we're going to try to do. The strategies were all developed by Mark Smedley, and I'll show you a picture of Mark in a minute. So this is the first shot that, sorry, I know this is not really, I'm not meant to show complicated things in presentations, but these are the vectors we used for gene editing, for the deletion of the DELA domain. And we were delighted that it worked with extremely high efficiency. These are the guides which make the DNA nicks on each side. And this is the big missing piece of DNA which used to encode the DELA domain. It's in frame, so now the start codon here runs through and just produces that next GRAS domain, which is the growth inhibitor, and in theory should give you a semi-dwarf or a dwarf, maybe even phenotype. So it was we were delighted to see. So I presented this at PAG in January. We just got this result, where indeed those lines with the DELA domain deleted are dwarf. This is the landrace that they're edited in, as usual, touching the lights in the controlled environment room. And these are the dwarfs next to it. So that's absolutely fantastic. For us, it was academically perfect. But in a way, it's still that problem of this is not the allele that modern breeders use. It might have negative pleiotropic effects, might behave in slightly different ways. We really like to recreate the alleles that are used. And I should say this was done by Shadia, who leads the wheat transformation team, Mark, who did all that construct work and the construct work I'm about to describe, and Rajni, who really did all the work. This is a lot of embryo excision and transformation and edit screening. So here's those cytosine base editors I talked about. This is how they work with the nickase disabled, this cytosine deaminase, which is going to turn the cytosine into a T. This is unstable while you've got the mispairing. So the edited T is protected by the uracil DNA glycosylase, these incredible proteins that people have designed over the years. And this edits within a window. So this is the editing window. And in theory, within that window, any C is liable to become a T. And the one we want is this one to make a new stop codon. When we've done this in the past, and many other groups have found this, we only change the bystanders, not the target C, or even the changes are not C to T, they're just random base changes, which aren't very useful. But just with recent results, we're really delighted to see that, again, with high efficiency and precision, we're achieving that base editing. So this is the target change. This is sequencing of, we've only got T0 plants for these because they're very new results. But Mark and Shadia and Rajni are efficiently converting this C to a T, making a new stop codon. This, I think, is another bystander. It doesn't make a difference for us because it's a stop and then a start, and then we use a methionine here. And this other SNP you see is actually a homolog because we're amplifying up more than one genome here. We haven't got pictures of the plants for you of these because they're just tiny plants at T0. So another final, but a very high ambition one, and one that we, the high risk element of this work, which we didn't necessarily need or expect to work, but was the perfect result, would be to achieve these changes with prime editing. This is even more incredible molecular biology, where reverse transcriptases have been added to the disabled Cas protein, and you're going to read off an RNA molecule to introduce new changes. This is the vector designed by Mark. Again, riding on the shoulders of giants, this incredible development work by these teams over the years, developing these increasingly efficient vectors for achieving this. And to our absolute amazement, it actually worked at high efficiency for us. So this is without any bystanders or changes outside of the region. This is the production of the stop codon exactly as we wanted it. It's the same allele that was used in the UK. I showed you those yield increases in the 70s, but that Green Revolution gene that was brought in fact, from Chile via Oregon State and Japan when it was brought in for a breeding program by Vogel and Barlog, etc. So it's really incredible to me to see that that's been recapitulated in land races collected 100 years ago. And it gets rid of that problem I showed you about. So these are the prime edited land races, perfect RHT1 semi-dwarfs, and they're going to be the thing that we use to start our new pedigree. This slide just gives you a bit more insight into the efficiencies. So of 14 transgenic Watkins plants that we tested, 11 showed prime editing activity, and 5 of them were high prime editing activity, with more than 30% of the SNP calls, G to T, shown within the next generation sequencing that we performed to screen for those edits. So for us, this is great because we've passed what was probably one of the most challenging milestones in terms of re-beginning these pedigrees. And that's exactly what we're doing now. We're removing the transgene from these edited lines. So that there'll be genuine GE plants, and then we'll start the crossing and the selection. I've got enough time left to tell you a bit more about my strategy in that crossing program, where I hope it's coming. Now it's clear that what we had was AG2 and 5, but breeders just intercrossed them. And if you look at where these ancestral groups come from, you know, that's basically everywhere where we grow wheat. And they've adapted to very diverse environments over 9,900 years. They've accumulated all sorts of alleles and traits which were specific to those environments, the way people ate the wheat, the way they grew it. And we think we should maintain these ancestral groupings. So we won't cross between ancestral groups. We'll make a semi-dwarf in each of them, at least one semi-dwarf in each of them. And then we'll cross within groups. And we'll have them, although the major genes for the UK will be there, the red grain colour, the late flowering, the hard grain texture, because we want to focus on bread making, all the other genetic variation will be maintained within those ancestral group clusters. So AG1 really will only be crossed with the other Chinese wheat that it came from, but we'll select for, in our case, for UK environments, hopefully in the future for other environments too, if we can fund it. And we think in that way, the endpoints will be really quite distinct from each other. It won't be a homogeneous, you know, pool of germplasm. It'll still hold the structure of that T-SNE plot I showed you. And then the next thing is, what are we going to select for? We could be Roland Biffin and select for yield, quality, disease resistance. But actually, the things we're finding in the Watkins collection now could allow us to select for other traits. This is Maria Hernandez-Soriano. Her paper is just being put into bioarchives here, where she's looked at the microbiome of the Watkins collection. And she's shown in this case that if you compare ancestral group 1, that's China, with modern wheat, there's many genera of microbes which are specifically recruited in ancestral group 1, which are not recruited by modern wheat. We don't know what they do. We don't know why they're recruited, but the recruitment is definitely an active interaction between the crop and the microbes. So we're thinking when we begin our pedigree selection, we'll actually try to impose some other criteria apart from just adaptation and yield, so that we can shift towards addressing some of those problems that I showed you in, I can't remember, an early slide about the global challenges of wheat, to get some sustainability traits bred in. And one example, for example, would be in collaboration with the Crop Sustain project, funded by Novo Nordisk and led by CIMMIT, where we'd start to select for biological nitrification inhibition. So start to try to reduce the production of greenhouse gases from the soil and from the microbes, because the wheat plants are producing microbes which antibiotics, which inhibit the development of those microbes. So they're the kind of traits we think it would be really important to go from ground zero on in these new pedigrees. Another more recurrent question in wheat, and one that we'd like to contribute to, is the question of hybrid wheat. So most of the world's major wheat programs have tried to produce hybrids, and there's many different reasons why hybrids are not really a commercial proposition yet. And I'm certainly not saying that a lack of heterosis is the only or the main problem. But it does strike you when you look at this T-SNE plot that within most of those programs, they were only crossing within the brick colored area, if that, probably more often within this diversity space. Whereas obviously to maximize heterosis, you really want to be crossing on the extreme sides of this diversity space, the edges, to maximize heterosis. And if you produce a well-adapted to lines which are specific to these ancestral groups, I think they may effectively be new heterotic groups in wheat, and the hybrids might display a higher level of heterosis than we've seen in wheat before. So this is not our primary goal at all. It's really more to do with the previous slides on capturing the diversity of wheat and the global journeys of adaptation that it took and what we can incorporate into modern. But as an aside, I think we'll be producing the opportunity to run this experiment. Okay, I've been speaking for 39 minutes, I think, so I've just got enough time to say a lot of thank yous here. I've just put together two really big programs. So the wheat work was mainly funded by the BBSRC in the UK, and that's a long-term program that was instigated by Professor Graham Moore, who you see here is in the DFW and DSW, and it was his idea to say, let's work across institutions. And certainly the data I've shown you, and lots that I haven't been able to show you, couldn't have been produced by the John Innes Centre alone, or by Rothamsted, or the other institutions which are part of the Cross Institute Wheat programme. So I think it's been a unique springboard and platform to be able to achieve that. I think that's why we built the link with Shifeng Cheng and Aegis, and why we had those great results. You're seeing the names of particular scientists who've contributed a lot over this time. The breeding companies in the UK, the wheat transformation team at JSE, my own group, who were always at the centre of this, organising the work. Major collaborations from Christobal Wawi's team, especially on the haplotype analysis, and from Rothamsted, led by Malcolm Hawkesford and Andrew Rich. So they were really the phenotyping hub for the work that I've described to you. I'm going to say it one more time, that the germplasm is all available at the GRU. Go onto that website, and if you need any of this germplasm and want it, contact them. Things like the genotype files and the phenotype data are all on Grassroots, and Grassroots is hosted by the Earlham Institute. So please take a look. Okay, thank you very much. Shall I stop sharing, Josh?
Yes. No, I think you can keep sharing.
Okay.
Okay, I'm not sure what. Okay. Yeah, in case you want to refer to anything, we have plenty of time for questions. Thank you for a great talk. I will try to bring up. Let's see. Oh, question and answer. All right. Okay, so I'm just going to start from the top. We have a question. The question is, will future wheat have more productivity or something else? Are you looking for more productivity, or or other characteristics, I guess?
Yeah. So I think one thing we'd say in the, we're geneticists, and we're not the ones who should be dictating what future wheat looks like. So we're trying to show the art of the possible, show what can be done, and then it's really up to an incredibly complex value chain, a food system, and most importantly, the people who eat it, right, about what future wheat should look like. So but having said that, I'll express some personal opinions where it's yes and no. If I we work a lot with colleagues in Kazakhstan, and in Kazakhstan, the average yields are like 1.5 tons, 1 tons to 1.5 tons. That's definitely worthwhile increasing productivity yield in those environments. They're a major exporter, and I think it would be a good way of increasing global food security with a very minimal environmental impact. In the UK, we've got world record yields, world record wheat from DSV called Champion, which I can't remember, some ridiculous yield, like 18 tons or something. The inputs and the agrochemicals used to achieve that probably mean that in environments like that, you probably would want to focus more on increased sustainability of farming and profitability. Of course, increased yield does not always mean increased profit. And in the DSW program, for example, we do a lot of work on reduced tillage. That's a lower yield, but it's cheaper farming. And at the end of the day, there's multiple benefits to that. So sorry for the complicated answer. That's the best I can do.
No, I think that's great. Okay, I want to comment that you've really made remarkable progress with wheat transformation, especially being able to go after different strains and make that successful. And then all of the work you've done with prime editing and the cytosine editing. So a question related to that is, are there any plans to mutate the MLO gene for mildew resistance?
That's a good question. So we've got that breeders toolkit I told you about. On that's we basically vote traits through, half academics, half commercial breeders. And if it's a recessive trait that you need to knock out on all 3 genomes in wheat, at best the breeders are going to be polite and say, okay, we'll take a look. But really, it's a huge problem in wheat when we need to try to exploit traits which are triple knockouts. For obvious reasons, they cross that again within their program, and only 1 in 64 individuals are going to be selectable for all the other traits. So the only way you get around that is a little bit like I said with the Watkins collection. You've got to come to fixation in the entire pedigree so that it's not segregating. So they're really tough. And so my honest answer is I think not in wheat.
Okay. I'm going to skip, let's see. Okay, regarding the base editing work, are you aware of frequency of off targets? Do you get a lot? Have you noticed any off target?
Bystander,
Yeah.
not off with, so that bystander I showed you, obviously we sequence them when we screen for the mutants. What I didn't say is when we segregate away the transgene and then we go to production of the pedigree, we'll carry out whole genome sequencing of each of the lines that goes into the pedigree. And increasingly, the UK experience is that if you do that, then you will find fragments of backbone and off targets in the background. That's a fact. In those cases, I don't know if that's what we're seeing, and the very high level of accuracy, etc., might not be the case. But certainly, we'd recommend whole genome sequencing in comparison with the original transform plant.
Okay, that's very useful. Thank you. Okay, here's a question. Is one reason for crossing within the ancestral groups, is that partly due to possible linkage drag?
Not so much linkage drag. It's the reason we're doing that is because when we look at the allele, obviously the ancestral groups are genotypically defined, but you do see in them different take the flowering time genes. They've each got their own signature of alleles that has been used for adaptation in that ancestral cluster. So what we want to do is maintain that and kind of go with nature and say, okay, we'll adapt the way it evolved over those years. So it's more a question of physiology and breeding than genome structure and linkage drag, etc. It's to do with trying to say, okay, well, what was achieved over that period of adaptation that is good, let's maintain it.
Oh, that's very interesting perspective. Okay, next question. Has work been you mentioned on biomass and yield components in the Watkins material been published? So I think you mentioned work on biomass and yield.
Yeah. So I sent Ying the reference list for this presentation. I'm sure she'll share that. And if you look at the Nature paper, the 2024 paper, then I really encourage you to dig into the supplementaries and additional data. It'll take you a while to navigate it, but in there you'll see the analysis of biomass. Of course, biomass is a low heritability trait. It's not easy, but it's there, and there were some interesting effects for it.
Okay, here's a practical question regarding the growth habit, the NAM populations that you've put together. Are the NAM progeny, are they spring type?
On the vast majority, yes. So it's a spring wheat times spring wheat cross.
Mm-hmm.
Of course, when you get a different VRN1 allele segregating, you get some winter type segregating out of that. But actually, I think, casting my mind back, I think the single seed descent was all without vernalization, so they'd all be spring.
Okay,
There's only about 80 winter types in the whole Watkins collection.
Oh, okay. Regarding your CRISPR methods, how do you stop Cas9 activity? I guess in the method, I guess you need it and then you don't.
Really good question, especially because some of those GA and sensitivity genes, if the edit gets too efficient and we start editing on all 3 genomes, you achieve sterility, which is not a good thing, hard to deal with. So to get rid of the at the moment the Cas9 activity, the only way we can get rid of it is through segregation. So obviously it's a different locus to the edit itself. You hope it's on a different chromosome, and then you just make a population screen for the transgenic backbone and the edit, and then you're looking for the ones which are only edit and not backbone.
Let's see, I'm kind of going through. Okay, so how can you maintain an increased genetic gain while facing challenges from climate change?
Well, first of all, you can, as I said in one of the last slides, you can stop contributing to climate change. I think that's an important thing that we need to start thinking about in agriculture and breeding. And one way, I mentioned BNI, so that's to reduce nitrous oxide production, and to some extent less production of fertilizer through Haber-Bosch. But in terms of it continuing to achieve gains in climate change, it's another one of those complex stories. And it depends which element of climate change you mean. In the UK, you know, the increased heat that we're getting so far is not detrimental for wheat yields. It's actually bringing some benefits. It's not going to be the case for other environments, I know. And you know, if you look at the success of CIMMET, for example, in breeding for heat and drought tolerance, I'd give a plug here for organizations like AHEAD within the Wheat Initiative. And my answer to breeding to adapt to heat and drought is to start getting our act together and collaborating internationally. And the best way I can see of doing that at the moment is to engage with AHEAD. I'd recommend to everyone, yeah.
Okay. All right, we have about 1 minute left. There's still quite a few questions. We're going to ask Simon if he can answer these offline and make his answers available. There was a question. We'll just make this the last question on nitrogen use efficiency. One of the audience members is curious about the numbers of QTL that you may be seeing for nitrogen use efficiency.
So they were from NUE and its components.
NUE, yeah.
Yeah. So they would be. We looked at NUE both in terms of classic NUE with nitrogen uptake efficiency times utilization efficiency equals use efficiency. So they're independent QTL for those 3. But also at Rothamsted, they actually looked at nitrogen partitioning within the canopy. So where is the nitrogen being partitioned in the canopy itself, or remobilized to the grain? So if you look at the QTL tables, you'll see all of those traits in there. And then finally, thing we would include in that, and in fact, one of the traits that we think has got most traction for at least more efficient use of nitrogen, if not nitrogen use efficiency, is GPD. This is grain protein difference. So, minimizing the dilution of protein when you get the yield increases.
Okay. All right. I think we've reached the end of our time. I really want to, let's all thank Simon Griffith again for a great webinar. If we could all clap. And I also want to thank all of the audience who joined us from all around the world for this webinar event. Thank you all for attending and for your interest in the topic, and we appreciate it, and we'll see you for the next webinar. All right.
Thank you.
Have a great day, everybody.
Bye-bye, everyone.
Thank you.